What if your cells couldn't properly recycle certain sugars, and that silent failure led to brain damage, skin abnormalities, and progressive disability? That's the reality of alpha-N-acetylgalactosaminidase deficiency, also known as Schindler disease, a rare inherited lysosomal storage disorder caused by mutations in the NAGA gene. This gene encodes the enzyme alpha-N-acetylgalactosaminidase, which is essential for breaking down glycoproteins and glycolipids inside lysosomes, the cell's recycling centers. Without this enzyme, partially digested sugar-containing molecules accumulate inside cells, particularly in the brain, nervous system, and skin, leading to gradual and sometimes severe tissue damage. There are three clinically distinct types of Schindler disease. Type 1, the infantile form, is the most severe. Symptoms appear within the first year of life and typically include profound muscle weakness, hypotonia, loss of motor and cognitive skills, seizures, spasticity, and early neurodegeneration. It often leads to death in early childhood. In this form, enzyme activity is either absent or nearly undetectable. Type 2, also called Kanzaki disease, has a milder, later onset presentation. It usually appears in adolescence or adulthood and is characterized by the presence of angiokeratomas, small, dark red skin lesions, along with mild peripheral neuropathy, muscle weakness, and in some cases, mild intellectual disability. Unlike the infantile form, some residual enzyme activity is typically present, which explains the less aggressive progression. The intermediate type features symptoms from both ends of the spectrum and may appear in early childhood or later. Neurological symptoms, such as developmental delays and motor impairment, are present, but may progress more slowly than in the infantile form. Schindler disease is inherited in an autosomal recessive manner, meaning a child must inherit two defective copies of the NAGA gene, one from each parent, to develop the condition. Carriers typically have no symptoms, but can pass the gene to their offspring. Diagnosis is based on a combination of clinical presentation, reduced enzyme activity in leukocytes or fibroblasts, and confirmation through genetic testing. There is currently no curative treatment. Management is supportive and may include physical therapy, anti-seizure medications, and treatment for skin or muscle symptoms. Genetic counseling is recommended for affected families, especially when considering future pregnancies. This content is for educational purposes only. For more science-backed explanations on rare genetic conditions, metabolic pathways, and human biology, subscribe to our channel and follow our educational series.